We're continuing now with the gram-positive cocci, and we're going to start discussing the Streptococcus genus. The Streptococcus genus is in the family of Streptococcaceae, and the various genera within the Streptococcaceae family include Streptococcus, Aerococcus, Leuconostoc, Gamella, and Pediococcus. There are also Streptococcus-like organisms. They look very similar to Streptococcus under a microscope. So the Aerococcus is a gram-positive cocci that tends to form tetrads, and if you remember from our early lectures, the tetrads are a group of 4 cocci. Aerococcus are alpha hemolytic and resemble the viridins group of the Streptococci organisms. These organisms may be confused with Enterococcus in biochemical reactions. Some other Streptococcus-like organisms, the Leuconostoc, again resemble Streptococci microscopically. On agar plates, the colonies resemble the viridins group of strep or Enterococcus, and these organisms are normally found in plants, vegetables, and dairy products. The Gamella, again similar in morphology and habitat to the viridin strep. The Gamella are easily decolorized with gram staining, so you have to be very careful because they will appear to be gram-negative cocci when in fact they're gram-positive cocci. And Gamella have been isolated from cases of endocarditis, wounds, and abscesses. Pediococcus are found ubiquitously in nature, and these organisms are commonly used to preserve foods such as cheese, meats, and vegetables, and these are rarely seen in human infections. The main species of Streptococcus that we're going to focus on in our clinical microbiology course is Streptococcus pyogenes, and this is also known as Group A Streptococcus. Streptococcaceae galactii, which is also known as Group B Streptococcus, the viridins group of Streptococcus, Streptococcus pneumoniae, and the Group D Streptococci, which include Enterococcus and the non-Enterococcus Group D organisms. Streptococci are classified based on several different properties. One is their hemolytic properties. Another are their antigenic properties. Streptococci produce Lancefield antigens and also some M proteins, and you can classify them based on these properties. And there are also biochemical properties like we test for every other organism. We have biochemical tests to differentiate these Streptococci. So I said the streps are commonly differentiated based on their hemolysis. So we mentioned previously that sheep blood agar, or what's called blood agar plates or BAPs, are commonly used in the clinical laboratory, and they're a great media because almost everything will grow on them except for a few fastidious organisms. So this slide shows the alpha hemolysis, where you have a greenish discoloration around the colony, and that's due to incomplete hemolysis of the red blood cells. Again, some pictures of alpha hemolysis where you get this green tinge anywhere where you have organism growing. Beta hemolysis or complete hemolysis, again, if you look around the colony, it's going to be a completely clear zone of hemolysis. Here's beta hemolysis. The best way to test for hemolysis is you take your plate, you hold it up to the light, and you want to see if you can really see through that zone around the colony or not. And then gamma hemolysis, of course, is no hemolysis at all. Streptococci have antigens on the cell wall, And you can group streptococci based on these Lancefield antigens. So you can group them from groups A all the way out to group O. For definitive identification, you can look at carbohydrates. Now, almost all of the streptococci have these Lancefield antigens on the outer surface, except the viridins group. The viridins group don't have any Lancefield antigens, so they're categorized as their own group of Streptococcus, and they're called the viridins Streptococcus. You can identify these Lancefield antigens on the surface of Streptococcus in various methodologies. So one of the older methods was to extract the cell wall antigens off the Streptococcus cells using a heat source. You can do fluorescent antibody staining, and the most common method for identifying Lancefield antigens is using an antibody-based coagulation reaction where you're looking for clumping in order to see what group you might be looking at. The most common groups isolated from a human sample are group A, B, D, F, and G. We already said that Streptococcus contains M proteins, and these are more common in the group A Streptococci. You can do precipitin tests to look for M proteins. The antibodies against M proteins are protective. Again, you can do biochemical properties in place of some of the immunologic antibody-based tests, and the biochemical properties are the more common methods used to differentiate the streptococci in a clinical setting. This is a chart which shows the different genus and species, their hemolysis, what Lancefield group they're in, the common terminology utilized, and the disease associated with each organism. This is a very nice chart and one that you might want to commit to memory, because questions like this, based off of this chart, being able to identify either the group or the genus and species and associating it with a specific disease for a case study is going to be to your benefit on an exam in this course, as well as on your Board of Registry exams that hopefully all of you are going to take when you complete this program. The cell wall of Streptococcus, because it's a gram-positive organism, if you remember, there's a thick peptidoglycan layer on the outer surface of the cell wall. There's also teichoic acid, but a much smaller amount of teichoic acid than peptidoglycan. There's a carbohydrate layer, that the Lancefield antigen carbohydrate layer, except for the viridins group. Remember, they don't have Lancefield antigens. And Streptococcus pneumoniae, a specific species of strep, does produce a capsule. And so that is one of the features, virulence factors of Streptococcus pneumoniae, that we will discuss when we talk about that organism. So this is the end of the first part of 4 parts of the Streptococcus lecture.